Genomics is the study of the complete genetic sequence of organisms. The Human Genome Project started in 1990 and was a 13-year effort that resulted in the sequence of the entire human genome. Sequencing technology has vastly improved since then. Many organisms have now been sequenced. Knowing the human genetic sequence opens up many possibilities for studying genetic diseases such as some cancers. All of the DNA inside each of your cells is about 5 feet long and contains approximately 20,000 genes. Researchers were surprised to find that most of your DNA does not code for proteins. About 98% of your genome does not get expressed as proteins. Large amounts of DNA are introns. These get cut out of our RNA and are not expressed in proteins. Transposable elements are capable of moving to different locations and make up about 44% of your genome. Another 44% is made up of repetitive elements, such as telomeres that stabilize the end of DNA strands. Historically, a gene was just considered a specific location on a chromosome. But with the discovery of so many different types of functions for DNA, it has caused us to redefine what we mean by a gene. In 2007, a new definition of gene was written as a gene is a genomic sequence, this can include DNA or RNA, that codes for some functional product. So this could be RNA or proteins. Now that we know the nucleotide sequences of many different organisms, we can compare them with humans to examine evolutionary relationships. Classification of living things is constantly changing as we determine more and more organisms' genomes. The x-axis on this graph shows how many nucleotides are present in the DNA. Essentially, this is how much DNA is present. The y-axis shows the number of genes. These are the sections that actually code for products like proteins. Humans have a little more than 3 billion base pairs, but only about 20,000 genes. Chimpanzees, our closest relative, are very similar to us in size and number. We share about 98% of our genome with chimpanzees. Some bacteria and yeast have very small genomes with relatively few genes. A nematode, which is a type of worm, has a very small amount of DNA, but about the same number of genes as humans. A common lab rat has a smaller genome than us, just under 3 billion base pairs, but actually more genes, about 25,000. Functional genomics is the study of the genome to discover what each gene does or what it produces. We use a tool called microarrays to compare the genetic sequences from different tissues. For example, we could compare a tissue that is not cancerous to one that is cancerous to try to determine which genes are related to having that type of cancer. Just like genomics is the study of genes, proteomics is the study of proteins. Proteomics studies the structure, function, and cellular interactions of proteins. Each cell produces hundreds of different proteins, so this is a huge area of research. Bioinformatics is the use of computer software to analyze genes and proteins. The genetic sequence by itself is just a huge number of data points. Computer tools such as BLAST are what are used to make comparisons to learn new things about these giant data sets.